Welcome, and my name is Luis Torres. This is a comparative network analysis of the mitochondrial recruitment of DRP1 for systems biology. Systems biology is an approach to understanding the nature of complex biological systems that are represented by networks. Networks consist of nodes and edges that represents interactions within a network. These networks can be analyzed to identify network characteristics in order to interpret possible patterns, help make comparisons, and predict outcomes for further research. This project applies these concepts on a type of cell death. Cell death is the irreversible terminal end stage of a cell that can no longer maintain its foundational life functions. A cell death, depending on the cause, can be uncontrolled or controlled. There are different process modes to a cell death which are interrelated through their interconnected and overlapped signal pathways, including but not limited to apoptosis, pyroptosis, necroptosis, and autophagy. This presentation focuses on apoptosis, and our current comprehension of apoptosis is that it is highly regulated and involves a signaling cascade. This cascade response is precisely regulated and is stimulated by pathological and physiological signals that the cell receives and responds to. Apoptosis is induced by events such as embryonic development where unwanted cells are shed, aging where old cells can no longer divide, or as an immune response to cell damage caused by injury, infections, or exposure to radicals. Apoptosis normally happens to worn-out cells, which calls for maintenance and the need to make new cells. Lack of apoptosis is cancer, while too much of it causes neurodegenerative diseases like Parkinson's disease, Alzheimer's, and Huntington's disease, for example, all which we still fully don't understand. Insufficient and excess apoptosis may also result in chronic inflammations and strokes, respectively, and a number of other diseases. The dynamin-related protein 1 promotes mitochondrial fission and localizes potential sites of organelle division, which is a part of the signaling cascade that causes for the mitochondria to swell, a key characteristic of an apoptotic cell. And analyzing the recruitment of DRP1 as a part of a network will allow us to observe how cell processes demonstrate evidence of function, distinct motives, and structured organizational properties. A network that is scale-free will follow a power law, meaning that the network does not have a characteristic scale and will have nodes that does not share particular sizes. This means that many nodes will have few edges or connections, while a few nodes will have many. Those nodes are called hubs. Small world networks are characterized by clusters and short average path lengths. Introducing randomness into a regular network can make it into a small world network, which creates shortcuts throughout networks, enhancing modularity and robustness through the connections of key hubs to local clusters. The hypothesis is that the DRP1 network will have properties of being scale-free and a small world network, and the randomized network will lack some of these properties. To test this, network statistics and motive enrichments of the networks were compared using Cytoscape and the analysis tool available. Reactome.org is a curated database of biological reactions and pathways. I chose to download from programmed cell death, went to apoptosis, and the apoptotic execution phase, more specifically the Homo sapien pathway for mitochondrial recruitment of DRP1. Then I imported it to Cytoscape as a network from file. From there, I can gather the summary statistics of my network by clicking Tools, Network Analyzer, and choosing to analyze my network. I click the default undirected network. A result panel displayed on the right side of the screen displayed key metrics that you see here. My chosen network notably lacked clusters in contrast to a typical regular biological network. It also has a characteristic path length of 5.26. I chose to randomize the same network in order to have a valid null model, to which I'll have a baseline for comparison and a controlled framework. This way I could see potential signs of an incomplete data, or something else that I might not be immediately taking note of to explain this anomaly. I did that by simply using Cytoscape's randomizer application. The randomized network is shown to the right. Then, with the side panel, a histogram plot can be created to analyze frequency of degrees in a network, which tells us the number of connections that a single node has to another node. The DRP1 network shows a sharp peak at degree 1, where 116 nodes have only one connection. It suggests sparse connections in the network and that it is hub-dominated and robust, where many nodes connect to a central hub or a small number of highly connected nodes. Though it lacks clusters, it is modular, and the relatively short average path length of 5.26 shows hierarchical structure, where hubs have peripheral nodes that do not directly connect to each other. The random network, on the other hand, is more uniform in structure and does not have highly dominant hubs, making the network less efficient and less robust. The path length is slightly longer and also lacks clusters. It suggests a lack of preferential attachment and functional specialization. So although the DRP1 network lacked clusters, it shows a centralized and hierarchical structure, good for more efficient protein-to-protein communication. So why were there no clusters? The explanation for why the DRP1 network lacked clusters, which are typically prominent in biological networks, is that the mitochondrial recruitment is involved in direct signaling cascades, rather than diffusing local networks like other broader cellular systems, for example. It directly reflects its functional design, which still supports the hypothesis, with hubs allowing efficient communication regardless of this lack of clusters, where the nature of the interactions are non-redundant, and the signal instead triggers the next from a central hub. A sign of evolutionary design, maybe? Interestingly, the summary statistics from the mitochondrial recruitment of DRP1 deviates a little from the expected characteristics of a cell network that involves a blend of scale-free and small-world network properties. The DRP1 recruitment represents a more specialized pathway rather than a clustered network, which highlights its unique role in mitochondrial fission. Despite these differences, comparing it to a random network allows for the consideration of the complexities, which should not be undermined. For future directions, it will be interesting to explore the DRP1 assembly and maybe investigate how scientists are now using live cell imaging and computational modeling to map mitochondrial recruitment of DRP1 relative to other apoptotic events. These observations help us be more informed about why disruptions in these processes can lead to various diseases that I previously mentioned. Thank you so much for watching.